All right. Morning, everyone. Thanks very much, Marco, for the introduction. A bit embarrassing. Yes, I'm at UCL, London, and I recently launched myself into a new adventure, setting up a lab in Rome, part of this Italian Institute of Technology. Let's see how it will work out. So, very happy to be here. Disclaimer, I'm not going to say much about imaging, so don't hit too hard about the technical aspects of imaging. I'm going to talk about what my group has been up to in the past 10, 15 years. The story starts with pain, but doesn't finish with pain at all. And I hope to provide a kind of a new perspective on this, which are the brain responses that we can record from a healthy human being when you give a transient nociceptive stimulus causing pain. These have been traditionally labeled as the pain matrix because of this natural tendency we have to associate the quality of the perception with the response we record from the brain. But I think I will try to show that those are actually reflecting a much more general property of the human brain, which is a very important one, which is the detection of important things happening in the environment in relation to action and to movement. So I want to start with a very basic thing. I don't know how many people here are working on like pain neuroimaging, but the very important thing is to ask a basic question. Why do we do this? Why bother running those experiments? And it's something that whatever you do is very important to ask yourself. And I think there are two different answers to this question. The first one, we want to understand how the brain works. It's an eminently physiological answer. And we want to understand the neural activity, the state of that system which gives rise to a certain perceptual experience, which could be pain or anything else you're interested in. This is not easy at all because, as I will show, these responses are somehow polluted by activities which are not related to that perceptual experience. And then there is a second objective, which is quite distinct, which is the objective to get a biomarker, to try to predict the intensity of a percept, in this case pain, but it's a general statement, the one I'm making, on the basis of a neural signal, on an MRI signal. And in this case, it's just a matter of decoding, and I'm saying just because it's a bit easier, and in a way, we don't care whether the activity, the signal we use, is something which is really mechanistically making the sensation itself. The problem in the field, and I think we can discuss this later, is that these two things have been conflated. And achieving two doesn't mean having achieved one. Getting a maybe useful biomarker, it doesn't mean that we have learned much about how the brain works with respect to the thing we want to measure. So when we give stimuli, nociceptive stimuli, like a laser zap, for example, or mechanical pain, we get very big responses in human brain. Using MRI, we get an array of brain areas, which has been described by many labs in many years. Has been so-called, said, the pain matrix. And if you record EEG, you get, after the stimulus, you get a very large biphasic response, an evoked potential, or as people say, an event-related potential. And this is dominated by these two waves, which are called vertex waves, because the scalp distribution of this signal, which is sampled at one electrode at the top of the head, if you look at these time points, they have a very centered around the top of the brain and widespread activity. So they're called the negative and positive vertex waves. And in some clinical fields, especially, they've been called pain evoked potentials. So to really understand what these things mean, I think it's very important to take a step back and say something which maybe might appear naive to you, but I think is important to keep in mind and ask basic questions about biology, because at the end we want to understand the biological system. So the couple of concepts I want to remind you is that are the following. That a key duty, if you want, of living beings is to replicate and reproduce. And the reason is that because reproduction produces that variability of phenotypes upon which natural selection acts and therefore allows the evolution of life. Keep in mind that as a final objective of living things, There is also something else to keep in mind, which is a prerequisite of reproduction, and that is survival. Because if you want to reproduce yourself, there is one thing you need to do, which is not to die, in order to be able to pass your genes to the next generation. Survival, reproduction, and evolution are the three main ingredients of life, and we need to keep that in mind when we study biological system. And also we should keep in mind that we study the brain, we measure activity from the brain, but we need to ask ourselves why there is a brain in first place. There is only a minority of biological entities with a brain, and those are things belonging to the animal kingdom. Those things which are alive and move in the environment, they have locomotion. And the only reason why we have a brain is because a brain is needed to cope with the world, with changes, sometimes very rapidly, sometimes unexpectedly, and in order to have like perception and action to achieve those ultimate goals of survival and reproduction. For these reasons, brains of different species have some innate knowledge of the world in which that species has lived. And the important thing about these innate models is that they don't like, they're not needed to have a faithful representation of reality, but they're needed to highlight things in the environment which are particularly relevant and salient. And there is a beautiful quote from a book by Richard Dawkins, which I think is very powerful, saying that if you have like a hypothetical zoologist with the tools to reconstruct and study a brain of an unknown species just by looking at that brain, he should be able to guess and infer the statistical properties of the world in which that animal and that species of animal has lived. So this innate knowledge of what in the environment is important, what is dangerous, what is necessary, is an innate, something very ingrained in brains. And brains have been shaped to select and highlight things in the environment which are very important to drive behavior, save your life, and pass your genes on. And if you think about this idea of brains having the need of detecting important things, well, there is a very important thing, which is the ability to detect what is dangerous and how to react to it. When I'm crossing a street, I want to have a system which tells me if there is a car coming fast and suddenly towards me. What about pain? Pain is a special case of this general principle. Pain exists, is there, is unpleasant. So, was a question? No, wasn't. What's happening? Nothing. Okay.
Just to test your.
Just to test my. Yeah, exactly. So it's important because it happens before there is an injury or before the injury becomes more serious. It's very useful from an evolutionary perspective. And in my lab, we have been trying to understand the rules behind the decision of the brain to detect these threats in the environment. And we have been seeing, for example, that these like models for detecting threat are very much dependent on the modality of the stimulus. Obviously, noxious, nociceptive stimuli are by definition threat, but they're not the only stimuli signaling a threat. Also the intensity of the stimulus, the time profile of the stimulus, and also, and this is where my research is going in a way, the position of stimuli with respect to where you are, what we call egocentric coding. And if we keep this in mind, I think it's easier to understand a bit the meaning of those responses. How do we get this? As I said, we activate the nociceptive system, for example, with a laser heat pulse. Nociception is the encoding of the system, and the elaboration of this afferent input in the spinothalamic system and then in the cortical projections is what produces the conscious sensation of, for example, pain. And those are the responses we sample from the human brain. What people tend to forget, and I think is behind what I'm trying to say, is that in addition to pain, the perceptual outcome of this activation of the system, there are other things as outcomes, which are sometimes neglected when we interpret our brain responses. And those are, for example, that you shift attention towards something that has happened. There are some autonomic responses, which I'm not very much an imaging person, but I think they are explaining quite a lot of what we see in imaging, especially in subcortical structures. And then there are, very important, some motor responses in order to act, do something with relation to that stimulus that has hit your hand, for example. However, these kind of things were a bit neglected, and the first imaging studies were focusing on this pain. There is a famous paper in Science, very old now, highly cited, describing the activity in those brain areas. And as you can see, the activity in those brain things, in those brain areas, was interpreted at that time as being, for example, in the cingulate cortex, surprisingly specific representation of pain. So it's very much biased towards the perceptual aspect of the outcome of that input. And obviously the pain center found in the brain and so on. And I admit that this kind of attitude has been now, is not as pervasive as before, but there are still a lot of things along those lines. It's certainly true that by looking at the amplitude of those responses, they correlate often, not always, with the intensity of pain perception. And on the basis of that correlation, for example, if you give a graded stimulus, you see a graded activity in those brain areas. If you do a regression analysis, or if you record ERPs, you see that the amplitude of the vertex wave, negative, positive, is related to the intensity of reported pain. And you can build some beautiful correlation maps for each electrode and skull maps in time, for example. However, correlation is not causality necessarily. And on the basis of that correlation, people have been inferring that those responses are somehow related to pain. And on the basis of that inference, people have been drawing and are still drawing, for example, clinical conclusions, like saying, if I have a patient in coma, I see activity in those areas, then that means that that person is experiencing pain and I need to treat that person. More recent work on newborns doing exactly the same kind of reasoning. In the field of social neuroscience, the assumption of those areas being producing the pain, somatosensory painful quality of sensation is used to understand, for example, things like empathy for pain. But what I'm trying to say is that these responses, which sometimes, not always, allow predicting subjective pain, and this both within subject, also between subjects, there are many papers coming out now. Are they really specific for pain? And the answer is quite simple. They're largely not specific for pain. And then there is a super interesting question, which is what are they for then? That correlation I was mentioning, which you can see here beautifully, graded stimuli, single trial ratings, then you sort your EEG responses according to perception. You see how they're beautifully graded, both the negative and the positive vertex waves. Well, that correlation can be disrupted. And in science, it's very important to try to falsify and break correlations. And this is something that we've done a few years ago. Instead of giving these stimuli like 10, 15 seconds, one from each other, if you start giving the same heat stimuli of 4 different energies, but in this way, so 4 energies, in little trains of 3 stimuli separated by 1 second, like pain, pain, pain. Now we've done many experiments up to 60 stimuli. And then you ask single trial ratings of the 3 pinprick sensations. Then you find something which is psychophysically, perceptually not surprising, that the intensity of pain depends very strongly on the energy of the stimulus, no surprise, and is kind of the same in the 3 stimuli of this little train. However, when you look at the brain response elicited by the 3 stimuli, the one elicited by the first stimulus is beautifully graded with intensity of perception, is that correlation. And here is key. The brain response caused by the second and by the third stimuli, which are as painful as the first one, they are very much smaller. And these responses lose the ability to predict your perceptual state. So it's an exception to the rule. This is really telling us that there is something more, that those responses cannot be made by neural activity, which is producing the pain itself. Otherwise, this correlation would not be disrupted. So in this case, we have the so-called pain response reduced with the same amount of pain. You can also have the other dissociation in the other direction. You can have this response without pain. And this is a paper that we published a few years ago, which was so hard to publish. Very simple experiment, a group of healthy subjects in the scanner receiving the laser pain on the foot, then receiving a very strong vibration, always on the foot, then some very loud auditory stimuli, some bang, and some very bright flashes of light, LEDs. 4 things which are perceptually very different, or equally surprising in a way. If you look at what happens with standard, simple, stupid, univariate GLM analysis in the brain in response to these 4 things, this is what you get. And usually I present this slide and I like to ask people, tell me which one is the sound, which one is the visual, and so on. And you can guess if you start looking carefully. Obviously here there is visual cortex. If you look carefully in the sagittal slices, here there is a blob in the sensory motor foot area, which is not present there. So we can probably say that this is something that has made contact with the body. But it's very difficult to immediately guess, because the point is that most, not everything, most of what we see is common across the 4 sensory modalities, at least at the macroscopic scale of, for example, function MRI. If you have questions, I'm very happy to take questions during the talk. If there is anything which is unclear or something you're upset about, let me know. If we do a simple conjunction analysis, and we put in yellow the areas which are commonly activated, then you see that most of what we get is kind of shared across modalities. So basically you can get the same type of pattern, roughly, when there is no pain. Recently, with Tim Solomons, we published this paper in a couple of patients with this rare condition called congenital insensitivity to pain, CIP, showing that basically when you look at standard areas activated by pain in both this neurosynth mask and the group of control subjects, they're basically the same areas which are activated in these patients in yellow with congenital pain sensitivity. So they're totally devoid of any painful percept, and is another proof of that, of what I just showed with the other modalities.
So were this patient receiving any sort of sensory?
Yes, they were detecting the stimuli, but they were not having the painful quality of the stimuli. It's a NAV 1.7 channel mutation. I don't know very much about it, but it was useful as an example to get something which is detected but without pain. It's kind of this thing looks using an abnormal trick.
You guys have estimate rate
yes.
The sensation. What were they reporting in terms of intensity?
They were reporting no pain because this is not, but they were detecting the stimuli in terms of intensity. So intensity was there, but not the painful thing. It's super interesting to look at what is really different between these two. And I'm not saying that it's the same, but the point I'm making is that at a general scale, there is most of what we get of the signal is shared. With EG, that big vertex wave, the same. If you do like the response elicited by sound, light, touch, and pain, you get always this vertex wave. I have a slide later. And you can see that this vertex wave, it's mostly contributed in yellow by activity which also contributes to the same vertex wave when it is elicited by sound, light, and touch. There are only a few things which are specific for somatosensory in the time domain. I'm not talking about brain oscillations, that's another story, very interesting. And this is the S1 component source of that electrophysiological response. The interesting thing, and here we get, I think it's something that really important, what is making these things? If you look at the contribution of the supramodal, as I call them, areas, the areas activated by all stimuli, at within subject level, what we found is that there is a convincing correlation between how much this multimodal activity in yellow contributes to the response and how much the subjects rated the stimulus salient. What does it mean, salient? It's a term I don't particularly like. It's probably one of the best things to capture what I think we want to capture, which is the ability of the stimulus to attract attention and force immediate behavior. Well, if we do this, the vertex wave, you can see that the contribution of these supramodal components is related to the subjective ratings of stimulus saliency across subjects. So you exploit the variability between subjects of ratings. Each dot is a subject, one modality. For example, this guy rates the visual very high, and in that guy, the contribution of supramodal things to the visual ERP were very high. These are the sources of the supramodal components, which are typically modeled with all the issues about source modeling, with a central dipole in the cingulate cortex and two bilateral dipoles in the operculo-insular cortex. And the same you see if you look at the supramodal areas in functional MRI, for example, the cingulate cortex or the secondary somatosensory cortex, which, despite the name, responds to auditory stimuli and to visual stimuli. And you can see how the activity in those areas is also related in a reasonably convincing way to ratings of salience. So before we move into the interesting bit, which is to understand what this is, the point, since I've been discussing this with Marco earlier, is that what has been called the pain matrix is something that you can actually get with stimuli which are not at all painful, if they are properly matched in terms of intensity and saliency. And because of this, we immediately understand that those kind of clinical conclusions, when I see that pattern of activity in the brain, it means that that person is feeling pain, is not holding, it's not valid anymore. It could be, but it could also be something else, and so on. And there are all these studies, old but also new in social neuroscience, looking at empathy, for example. This is one of the first ones, cyberball, people in the scanner playing with someone else and then being excluded. And when the person is excluded from the game, there is activity in those areas, and activity in those areas, because of the similarity, has been interpreted as being, it's been interpreted as being like, oh, it's really pain. It really hurts when you lose someone we love. And then there is another paper in which this has been done more directly, comparing the brain response to transient pain with a short presentation of a photograph of the former boyfriend or girlfriend who left the volunteer in the scanner. And on the basis of that overlap, yeah, you shake your hand, but you shake your head, but I mean, it's PNS, you want to publish how you go, these kind of things. The idea is that on the basis of this overlap, it was concluded that social rejection has the same somatosensory representation of physical pain. I think this is a bit far-fetched. It's fair to say that the same group published a few years later a new analysis using a fancier approach, MVPA, to distinguish between the two conditions, and they did find some differences, as expected. And then things become interesting. But in general, it's very easy to make mistakes. This is a typical, another example, newborns, healthy brain, so-called pain matrix. We've seen you can get this with auditory stimuli. Say, oh, I see the same in newborns. It means that they feel pain. So we need to treat them. Again, maybe we need to treat them, but this is not the evidence to make that conclusion. Sorry, this shouldn't be there. And obviously, this idea, maybe this is super naive for you, but it's what people like Poldrack have been saying for a long time. This difficulty of inferring perceptual state or a condition from a neuroimaging thing is, formally speaking, is a logical error, inferring like a perceptual state on the basis of brain activation. It's something that we do all the time in science, actually, when we have a measure inferring an underlying condition. And it's a bit saying, okay, it's okay to say if A then B, then I run my experiment, I see A, I conclude B. No problem at all. It's a bit like saying if someone does laundry, uses more electricity, then I run my experiment, I see that that person is doing laundry, and then I can be certain that he's going to pay more at the end of the month. Something similar, superficially but profoundly different, is the opposite. When he does laundry, he uses more electricity. I run my experiment, I observe that the electricity bill is increased, and then I conclude that, that person was doing laundry. It's obvious that there is a problem here. The problem is that there are other possible explanations for that observation. For example, that person was using another electrical appliance. And this is exactly the same thing that people do, and we do in MRI, in pain, in many things. When we say we give the laser, I see activity in those areas. And then when I see activity in those areas, that means that that person was feeling pain. Possible, but not certain. Depending on what? Depending on the exclusivity of the relationship between the activity in the brain and the perceptual state. So this has to be dealt with in a probabilistic way. And basically, we need to draw those conclusions, we need to measure how easy it is that those things are exclusively related. And the problem is that the response selectivity of those brain regions with the analysis techniques that we use is very low. And I'm sure you run MRI experiments all the time, and I'm sure you find activity in the insular cortex, in the cingulate cortex, almost whatever you do, if you're working on gambling or addiction or pain or you name it. And those are the brain regions most activated ever, like the cingulate, for example. This meta-analysis, negative affect, pain, cognitive control, all activating the cingulate, which is therefore telling us that the relationship between the measure and the underlying perceptual state is not exclusive. Therefore, when I see this, it's very hard to say, it means something else. And this is, for example, I was reading some papers on gravity perception, and people find activity in the insula. There is a high-profile paper saying, gravity is encoded in the brain in the insula, because with a gravity task, you activate the insula, and then they say, the insula is also activated by vestibular caloric stimulation, therefore this is gravity. But to do that, you need to test all possible other stimuli which are activating the insula. And if, and only if, you don't see that other conditions activate that brain area, then you can draw your conclusion. And this is an old review paper where we discussed this thing. So in terms of a bit of self-advertising, this is a big review that came out in December in Brain about this problem of pain biomarkers using neuroimaging. The problem, I think, and we can discuss this, that all the attempts to falsify the hypothesis that these measured brain responses are specific for pain, they're not being sufficient because people don't use the control stimuli which are necessary to be used. And then people don't use the word pain matrix anymore. They use pain network, pain signature. Signature is a difficult and dangerous word because it implies specificity. Or neural circuits, which are all in a way equally suggestive and giving that message of the response being related to pain. Okay, I think the fun part of all this, I could give a talk on the bits of the response which are specific for pain, but I prefer to talk about the bits which are not specific for pain, because this is, I think, in my opinion, is more fun, understanding what these things are. These yellow responses work. Was it your question? No. Is it clear so far? Okay. Well, a possibility, which I alluded to at the beginning, and if you think about what I said at the beginning is something that should be considered, is that this activity in the brain is the result of a feature of the brain, of a filter in the brain, which is able to identify things in order to orient attention and react to these things in the event which are important. If you consider the old René Descartes little man, the first description of the nociceptive system, the fire, before being a nociceptive stimulus, it's a very powerful visual stimulus. And you do want to have in your brain a system which is able to rapidly identify the fire, the predator, the car, which is going to hit you, and make that decision and do something motorically in order to save your life, hopefully, and be able to achieve the ultimate goal of passing your genes to the next generation. it's interesting that when you look at behavior of epileptic patients with electrodes in the cingulate cortex, the areas which are also activated in pain studies, blue are the place where the needle is. In some of these places, the very artificial electrical stimulation of the brain produces vaguely defined urge to move, urge to run away from something, although there is not anything to run away. So what I'm really passionate about at the moment is to understand this thing. So this big vertex wave was described for a long time. One of the best descriptions is from Giandomenico in France in the 50s. This thing is the largest activity that you can record from the brain of a healthy human being during wakefulness. There are bigger things during sleep. That's a different story. And still, we don't know why it is there. So why there is this big vertex wave? We know that sometimes correlates with perception, not always. We can disrupt that correlation. We know that you can get it in response to laser stimuli, pain, but also in response to visual, auditory, and tactile stimuli. Again, the distribution is pretty similar. And I'm very interested in understanding what are the rules behind the decision that the brain makes to produce such a massive thing. We're talking about 100 microvolts peak to peak on a single trial level. This is the largest thing we can get in the human brain in terms of transient synchronization of thalamocortical activity. And then keeping in mind that idea of like the motor consequences of having a brain, then we run, oh, sorry, I forgot. This is just to show that by playing with this experiment with the little trains of these stimuli, showing the habituation of that vertex wave, if instead of giving always the same stimulus at the same intensity, in the same position, you play around with one by one with these features. For example, you compare pain, pain, pain with sound, sound, pain, and you look at the response caused by these two, which are identical. The only thing that changes is the context, because they're preceded by two sounds. Well, what you see is that the brain has a filter to detect that the change happened. So the change in modality is what I referred to at the beginning. It's very important. It's something that drives the brain to produce that big thing. Intensity, there are interesting rules here. Increases in intensity are detected. Decreases in intensity are not detected. And then when you look at space, becomes also very interesting because we have some results showing that, for example, if you give the three stimuli here, for example, on the left hand, and then you compare the same stimuli on the right hand, left hand, and we look at this subject in front of the table, the response caused by the third stimulus is the same. So the brain doesn't seem to be tweaked or tuned to detect those kind of spatial changes. But then if you increase the somatotopic distance between, for example, left hand, and here is left hand but preceded not by right hand but by right foot, then you do get a smaller dishabituation, smaller than change of modality. And the interesting thing is that obviously here the stimuli, left hand, right foot, are distant, both in somatotopic coordinates, in body map coordinates, but also in egocentric coordinates, in Euclidean distance between the two stimuli. So if you repeat exactly the same condition, in which you have right foot, left hand, but then you ask the subject, instead of like sitting like this, to put the left hand and the right foot close to each other. So it's a nice way to dissociate egocentric distance versus somatotopic distance, then that dishabituation is gone. So it seems that there is a complex set of rules behind the decision of the brain to make this big vertex wave. Oops, what happened? Gosh, that's the nightmare of any speaker. Okay. So let's see. So in order to this is a recording.
Yeah, don't worry.
We need to get back to, okay.
Sure.
So what is this big thing? And then we run a very simple experiment in which we wanted to test whether that evoked potential has an effect on the motor system. The experiment is super simple. The subject is holding a force transducer, so we measure force. Look at me, don't look at the slide. And the only task, it's very simple and boring, is to hold the transducer at a certain constant force. And then we give the stimuli, like somatosensory stimulus, pain, or an auditory stimulus, which we know produce the big vertex wave. And we looked at what happens in the force. Well, the subject notices nothing, but when you look at the force, give me a whiteboard, you see that after the stimulus, I was expecting either to find nothing or to find an increase of force or a decrease of force. You actually find a three-phasic modulation of the exerted force. Is this the slide now? Okay, go. Well done.
The right place?
I think so.
Okay.
Kind of. Okay. So you measure this force with this trick. And then you get the time course of force. So the subject is there, gets bang, shock. And then we measure force, either auditory or somatosensory. And this is what you get. So time of the stimulus, if you baseline correct, of course, then you get these three different modulations. There is a first drop, about 100 milliseconds following the stimulus, then an increase of force, and then a longer lasting increase of force. This is a t-test to show how consistent it is across subjects. It's a very small thing. We're talking about 2%. The subject doesn't notice, but it seems that those kind of stimuli, which are judged by the brain important enough to produce a vertex wave, are related to a modulation of the motor system. Wow, and a complex one. Then we did the same with EEG, trying to understand better the relationship between the two. And if you look, the same modulation of force, this is very reproducible if you want to try. It's quite easy. Then you get the big vertex wave, negative, positive, which we know. And it's very tempting to say that the two things are related, because somehow they happen at the same time. They seem to have changes of polarities which are similar. And then what we did here is a bit complicated, but I hope to explain what we did is to look at the variability between this vertex wave from trial to trial and the variability of the force from trial to trial. So we have brain activity here, negative and positive brain, and force modulation across time. And then we've done a correlation for all time points, looking at within subject variability between modulations of brain and force. One electrode, of course, and here there is only one electrode because one sensor. And then you see that there are many areas which are positively correlated between the amplitude of the positive peak, for example, and the increases of force. And obviously a negative correlation between the negative peak and the increase of force. This is within subject. Between subjects, you can do the same. Instead of entering with single trials, you enter with one average from each subject, and then you test whether people with a big vertex wave also have a bigger modulation of force. And then you do get the same here, negative correlation at CZ. This is a big, like a cluster-based permutation, so it's a quite a strict test. There is a trend of something happening there. But the interesting thing is, the positivity of the vertex wave or the negativity are able to predict on a trial-by-trial basis and across subjects how much the subject will squeeze the transducer unconsciously later on, up to 2 seconds after the stimulus. Then what you can do, you can look at these correlations in scalp array. Here is one electrode, but you can scalp map these positive correlations of the late occurring, time is going this way, P wave, and of the earlier negative. And what we see here, if you look at this positive, you see that they are ipsilateral to the stimulus side and contralateral to the hand holding the transducer. And if you look at this one, instead, they are contralateral to the stimulus side and ipsilateral to the hand holding the transducer. What does it mean? I don't know what it means. It's something that we're investigating. The only thing I know, and makes it interesting in my opinion, is that these things are lateralized. Those are correlation maps. We're plotting R or T. If you compare this with the topography of what the brain does, this is purely symmetrical. It's a vertex thing. So it means that probably this big thalamocortical activity is somehow affecting other things in the brain in order to modulate behavior. And remember how much I banged against correlations not being causality, and how with the train of stimuli paradigm, we showed, for example, that the pain, so-called pain responses are not pain. Therefore, we've done the same experiment. This time we're auditory stimuli, bang, 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 producing the well-known habituation of the vertex wave, okay? What do you expect the force to do? To follow what the brain does? To follow what the afferent input is? Well, we didn't know, and it seems to follow what the brain does. So that modulation, which breaks the relationship between perceived intensity and saliency response, is not breaking the relationship between the saliency response and the force, the motor output modulation. So just to recap, those are responses that, I don't know, people here working on ERPs, traditionally they're interpreted in the sensory domain. In the pain field, they were considered pain-evoked potentials or sensory-related responses. I think what we have found here is a basic physiological mechanism that couples these decisions of the brain of what is important with the modulation of the motor output following a complicated pattern, which it's nice to speculate that could be important for to drive behavior in a useful way. This is not a reflex. It really depends on those set of rules that the brain uses to decide what is important. And something that we are testing now, this is just a hypothesis, is that this kind of thing is probably related to the detection of, the execution of some defensive actions. How am I doing with time and how am I doing with your attention?
About 5 minutes.
Okay, so the last part, very simple. It's about reflex responses. There is a scientist who's surprisingly ignored, Ivan Sechenov. How many of you have heard about Ivan Sechenov? 0. Wow. He was the master, the professor of Ivan Pavlov, the guy of the dog, and he was having fun decapitating frogs and getting reflex responses. And he found that if you decapitate a frog, you produce these movements, and he said something which is revealing for me, that the objective of these movements is invariably to preserve the integrity of the body. And he found that if you decapitate a frog, so no brain, if you stimulate the frog in the water, on a table, the frog withdraw the paw, but if you stimulate the frog in the water, the frog pushes away the paw. And he was puzzled because he said, it seems like that there is a kind of intellect in the spinal cord of the frog, which is able to decide what to do in a purposeful way to facilitate useful behavior. And what he found, he said, it's very good if you want to record reflexes, cut the head, because you get better reflexes. But as soon as you attach a brain to that circuit, the sphere of reflex movement is immeasurably wider. And he got in trouble because he was one of the first one to say that psychical phenomena are, in effect, a machine-like process. And this idea of modulation by the brain of subcortical circuits of stereotype movement is something that we have done, recording a very simple defensive behavior, which is the blink reflex. When you close your eyes in response to a stimulus, you can produce this blink reflex in response to a shock to the hand, subcortical response happening entirely in the brainstem, very easy to record in the 2 eyes. And what we found is that 50 milliseconds, there is no time for the input to go up to the cortex. It's present in people in coma. What we found is that if you stimulate the hand, you get this response. Those are 20 trials in the 2 eyes, on the same side and opposite side of the hand stimulated. But then if you ask the subject to put the hand closer to the face, in other words, to put the stimulus closer to the part of the body the behavior is meant to be protective for, which is the eye blink, then despite the circuit is the same and the latency doesn't change, the response is massively increased by a factor 2, so it's doubled. Wow, interesting. How is it possible? Well, it is possible because, and I have lots of physiological data I'm not going to bother you with, that this increase of hand blink reflex, HBR, when the hand is far and when the hand is near, it's because there is a tonic modulation in a top-down fashion from the cortex, specifically of those circuits in the brainstem which underlie the production of the blink. So the cortex decides that the stimulus is close to the part to protect, and therefore it's worthwhile, it can be bothered to enhance the muscular response which is broken. And this idea was to, we started reading this literature about peripersonal space, coding of space surrounding the body, which is a very difficult concept to deal with because there is lots of terminological confusion in the literature. But the idea was that when the threat becomes closer to the body, the brain decides to enhance some type of motor behavior. And then by playing with different hand position, I don't want to bother you with this. So you use the hand as a locator of the danger with respect to this HBR, blink, defensive blink. Then you can estimate how this function or peripersonal space. Some people use the concept of a bubble to communicate. I don't think it's an appropriate one, although it's very easy, to measure how this changes in different people. And for example, you can see that the size of this, what we call, skipping this, sorry, what we call defensive peripersonal space of the face, if you look at the size of it, is related, for example, to the anxiety trait. So people who are more anxious have a brain which decides, even when the stimulus is farther away, to facilitate the circuit underlying that defensive model. And then by playing with different stimulus positions, you can do it with the hand, you can do it with auditory stimuli moving around. This is work that was driven by Chiara Sambo, who was with me in London a few years ago. Basically, we found that the idea is that the brain estimates the danger of things in the environment which can move to interact with you. And this is obviously the potential to harm of things in the world. So this corner is very dangerous for me. So I need to have a system which is able to decide that it's very important that I deviate or someone coming towards me. And obviously proximity is something which is unconsciously estimated by the brain to decide which kind of motor repertoire deploy in order to protect yourself better. And by playing with this position of stimulus in space and getting a measure, in this case the defensive blink, then you can do this. Oops, you can build these maps showing how big the response is as a function of the stimulus in space. You can see that it's symmetrically, right-left. It becomes asymmetrical in some pathologies. For example, in trigeminal neuralgia, chronic pain on one side becomes expanded on one side. It's bigger above than below, something we did not expect. And it's funny to see this because then you can test things. And why do you think you have more defensive response when the stimulus is up here than up there? Because on Earth, things tend to go down because there is gravity. So something up here is more likely to hit the part you want to defend than something at the same distance, which is down there. Then you can do a nice experiments building the same map when the subject is standing, and you get that kind of shape, or doing it when the subject is lying on the floor. And then you see that modulation function is always sticking up and gives priority of things which are falling from the top. So like physical laws are kind of ingrained in that innate knowledge of the world, which is embedded in brains, which I was talking about at the beginning. And I think I finish here, but the important thing I want to say is that this peripersonal space, which is often presented as a thing, like the peripersonal space, well, we just published an opinion paper in Fancy Cognitive Science. I think it's not exactly a space. This is just the assessment that the brain makes of some actions, in this case the blink reflex, but this happens for many other actions, as a function of their utility value. So it's not a defensive space, it's the value that the brain assigns to some to the relevance of some actions. And in fact, it's not only modulated by proximity, because if instead of using like a loudspeaker with a bang, I use a suddenly appearing knife versus a suddenly appearing cotton thing, then you get also some semantic modulation. So it's not just depending on proximity, it's really a global assessment of the value of things happening in the world and the consequent actions in order to protect yourself. I have one slide that is, I think, shows this idea, which is this a bit of self-advertising. This is the where we put forward this kind of concepts, and the idea is that is this one. For example, imagine that you have two actions we can measure from the brain. One is climb a tree, and one is run away when you are in front of a crocodile. Obviously, the relevance of these two actions depends on the position of the stimulus with respect to you. So you can plot the relevance of the two actions as a function of the position of the crocodile, and climbing the tree becomes super relevant when the crocodile approaches, because the crocodile cannot climb the tree, but goes back to zero when the crocodile is in between you and the tree. And at that point, the relevance of running away action becomes super high. If you do this, you can build some nice, as we call them, fields, defining the relevance of those actions, like climb the tree or run away. And people have been using these as measures of the peripersonal space, but actually there are so many measures because there are so many actions to be mapped. And it's all dependent on the context. So if instead of having crocodile and tree, you have no tree, and you plot the relevance of the same actions, well, the relevance of climbing the tree is zero, and you just have a field mapping the relevance of running away, which gets bigger and bigger when the crocodile approaches. If the crocodile is someone you're attracted to, there is no relevance of the action running away, for example. Okay, so take-home messages. I think I showed that brains have evolved to be able to cope with the world, that we and other species perceive the world through innate images of the world. Those are shaped by natural selection, and they're important to achieve the ultimate goals of survival and reproduction. There is a complicated system of hierarchical rules which to identify the behavioral relevance of things happening in the world based on different things. Pain, nociception is one very important factor producing that. And there should be, when we get these rules are met, then the brain bothers producing this big activity, which is, however, a mixture of things, so it's very heterogeneous and functionally different. Only a small part of it is related to the quality of the sensation. If we are interested in that, we need to use good control stimuli to tease out what is perceptually important. The large part of this is supramodal, is a large synchronization of neural activity. We don't know exactly what it does. We have preliminary evidence that is related to something having a motor output, because the idea that we have a brain just to move, first instance. And then I showed that even subcortical responses, reflexes, are in fact modulated in a very intelligent way by the cortex, defining things like a defensive peripersonal space, which should be more conceptualized as fields of relevance of actions. And this is the most important slide. People are working at the moment with me between London and Rome, some previous lab members. Giacomo has been behind all the force stuff. Rory, the modeling, Chiara, the peripersonal space, André Moreau and Liku in the pain matrix stuff. And I take this opportunity to say that I'm setting up this new lab in Italy. I have 3 or 4 postdoc positions coming up. They have to be filled this year. If there is anyone interested in coming with us and work on this kind of things with this kind of attitude, I would be grateful if you could let me know. Thank you very much.